this principles of inheritance and variation, right? So in this chapter already we saw about the monohybrid cross and the dihybrid cross and about Mendel's law of inheritance regarding the monohybrid cross and dihybrid cross, right? So, and also we saw about the gene interaction, that is the intergenic gene interaction. So examples for the intergenic gene interactions, we saw about the complementary genes, supplementary genes, and epistasis, right? And today we'll focus on the allelic interactions, which is also called the intragenic interactions, right? So under the allelic interaction, that is incomplete dominance, then codominance, multiple allelism, all these comes under the allelic interaction. That is the interaction between the alleles of the same gene. That is intragenic interaction. Okay. And also we'll see about the chromosomal theory of inheritance and what is linkage and recombination. So all these actually they are all the variants of the Mendelian law of inheritance. That is, they do not follow the Mendelian law of inheritance. So in what way they differ? So that's our main focus today. Okay, so non-Mendelian inheritance, right? So it is one of the interesting topics. So first we'll see about incomplete dominance. So what do you mean by incomplete dominance? We saw the law of dominance, right? So what do we see under law of dominance? That is, under law of dominance, out of the two alleles, one allele will be the dominating, right? The other one will be the, in the case of heterozygous condition, the other one will be recessive. And if the recessive character is to be expressed, it has to be in the homozygous form. That is, both should be in the same, right? So, but in the case of incomplete dominance, what happened? It is the inheritance in which the offspring shows intermediate character between the two parental characteristics, right? So here, what happened in the case of Mendelian inheritance, if you take the tall and if it is crossed with the short, in the F1 generation, we'll get the heterozygous condition, which is a tall, similar to that of the parent, right? But in the case of incomplete dominance, it is the inheritance which shows the intermediate character between the two parental characteristics. So here, when we cross the red and the white, red with the white flower, what happens is we'll get the heterozygous type, but this heterozygous type is not of the red color, but this is of pink. Okay, the flowers obtained will be of pink. So here the example is the flower color in snapdragon. So that is also called dark flower or antirrhinum species. So it is a well-known example for incomplete dominance. So the same type of inheritance as Mendel explained was not observed in all the plants. Okay, they also shows the intermediate characters. So what does this indicate? That is, this R is not completely dominant, but it shows the incomplete dominance. Because if it is completely dominant, if the capital R is present, it should be of red color. So it is not completely dominant over the small r. So when both the alleles are present, that is in the heterozygous form, it shows the pink color. And also another example is Mirabilis jalapa. That is also called the 4 o'clock plant. So these two are the example, and also it is observed in animals also. For example, in Andalusian fowls. In Andalusian fowls is an example for incomplete dominance. So here the feather color, for example, black, when it is crossed with white, it shows the F1 generation, it shows blue color. It is the feather color in Andalusian fowl, it shows blue color in the F1 generation. So here, in the case of snapdragon or the antirrhinum species, it is one of the common example for the incomplete dominance. So a cross between homozygous red and white, it produces a pink flowered plant. Okay. So these three are the main examples for incomplete dominance. One is snapdragon and mirabilis jalapa and Andalusian fowl. The feather color in Andalusian fowl. So these are the examples for incomplete dominance. Okay. So if you observe the phenotypic ratio, for example, so P, this represents the parental generation. If the capital P is shown, it shows the parental generation, right? So this is the parental generation and we are crossing the R, that is the homozygous R with the homozygous. Here the character is white, right? White color flower. So red color flower, that is pure generation of red, and this is the true bred line of white. So this is homozygous recessive. So when it is crossed, in the F1 generation, all the flowers are pink. That is heterozygous condition, it is pink. And these are the possible gametes, right? R and that is capital R and small r. And when these, that is the product of the F1 generation, this heterozygous R, it is crossed with the other pink flower, then we'll get it in the ratio of, so this we can draw it in the form of a Punnett square, and you can find out that it is obtained in the ratio of 1:2:1. So the phenotypic ratio is 1 will be red and 2 will be of pink and 1 will be of white. And even the genotypic ratio is same, that is 1:2:1. So this is same as that of the Mendel's hybrid cross, monohybrid cross, right? And this is, they are called Punnett square. Punnett square mega, that is where the Punnett square is actually used to show the graphical representation of the probability of the offsprings, right? So we can find out if the gametes are, that is, this is the gamete from one parent and this is the gamete from other parent. So we can draw in the form of Punnett square, that is, this will have R, that is both the capital, here this R, and here one is uppercase and other is lowercase, and when these two breed together, it will be R. So this can be represented in the form of Punnett square. Okay, so this will help you to find out the, so what does this indicate? That is the probability of the offsprings that we will get, right? So these are the probable offsprings that are obtained by the crossing between the parent, that is RR and RR, okay, both the heterozygous. So these are the probable offspring. Okay, so this is called the Punnett square. So when we compare the ratio, the genotypic ratio, even in the case of Mendelian monohybrid cross is 1:2:1. But the phenotypic ratio in the Mendelian monohybrid cross is 3:1, right? I hope you know this. So it is 3:1. But instead of getting the 3:1 ratio here, we are getting the phenotypic ratio of 1:2:1. That is, both phenotypic and genotypic ratios are the same here in the case of incomplete dominance. Okay. So what does this indicate? That is, R is not completely dominant over small r. That is, the capital R is not completely dominant over small r. That is, capital R in the sense red. The red is not completely dominant. So it is incomplete dominant. So what do you mean by incomplete dominance? That is, only when it is present in the homozygous form, it shows the red color, and when it is present in the heterozygous form, so even though this uppercase or the capital letter R is present, it is not showing the red color, but it is incomplete dominant and it shows the pink color. That is, there occurs an interaction between the two alleles and it shows the pink color. Okay. P plant also shows incomplete dominance in other traits. In some of the traits, even P plant shows incomplete dominance. So this is about the incomplete dominance. So here what I have to be clear is what is incomplete dominance and about the ratio. So here the phenotypic ratio, it differs from the Mendelian monohybrid cross ratio. Okay, instead of 3:1, we are getting 1:2:1. So this is the difference. And then the next non-Mendelian inheritance is codominance. So what do you mean by codominance? It is the inheritance in which both alleles of a gene are expressed in a hybrid. So if they are present in the form of hybrid, both alleles will be equally expressive. The example is the blood group, ABO blood grouping in human. So the ABO blood groups, it is controlled by the gene that is called the GI. So here, if you take the blood group A, they will have a special antigen which is called the antigen A. And if you take blood group B, they have the special antigen that is antigen B. And if you take blood group AB, they will have both antigen A and B. And blood group O, there will be no antigen. That is no antigen A or antigen B. So actually, what does this represent? That is each and every blood group, that is they have the sugar residues. That is this gene I, it controls the production of sugar polymers. That is they have some antigens in the plasma membrane of RBC. So this plasma membrane of RBC, they contain some sugar residues which are responsible for the difference in the antigen A or antigen B. So this gene I, it has actually 3 alleles which are represented as IA, IB and I. So this IA and IB, they produce a slightly different form of the sugar state. So they differ in their antigen that is present, the sugar in the type of sugar that is present. Okay, but this allele I, which is represented as small letter, right? It does not produce any sugar. So for example, this blood group O, so they don't have any sugar. So if you take the genotype of the different blood groups, you know that there are 4 blood groups, right? A, B, AB and O. So if you take the blood group A, that is people with blood group A, they have the genotype of either IA and IA. That is one IA is from one parent and the other IA from another parent, right? Or they may have IA and I. That is I from one parent and IA from another parent. So if IA and I are present here, the A is dominant, so they will express the blood group A. So A antigen, they will have A antigen, so their blood group will be A. Similarly, in the case of B blood group, they may have either IB, that is both B from, that is one B, this is from one parent and this is from another parent. Or they may have IB and I, that is IB from one parent and I from another parent. So then they will have the blood group B. So here the IB is dominant over I. So when B is present, they will have the blood group B. And in the case of AB, so they will have the IA from one parent and IB from the other parent. So in this case, it is called the codominance. That is both, they will have antigen A as well as antigen B. That is blood group with AB, they will have both A and B antigen. And those people have the blood group O, they have no antigen I. This is the only possible form. Okay. So based on that, there are 6 genotypes are possible. So 6 genotypes are possible. So when IA and IB are present together, they both express their own type of sugar, that is antigen A and antigen B. And this is due to the codominance. So here both are equally dominant. It is not that A is present, so A blood group. So here A is dominant. So here B is dominant, but here both are dominant, that is equally dominant. So that is called codominance in the case of IA, IB. Okay. So if you're getting the allele from parent 1, IA, and allele from parent 2 is also IA. So the genotype of the offspring will be IA. And the blood type, that is phenotype of the blood type, will be A blood group. So here, if IA from one parent and I from the another parent, it will be IA, and also the offspring blood type will be A. Okay, so in both the cases, A. Similarly, for B, IB or IB, I. It represents the blood group B. So if the offspring or the progeny is having blood group AB, they may receive either IA from one parent and IB from another parent, or IB from one parent and IA from another parent. Then they will get the blood group AB. And I, that is with no sugar or no antigen, and from another parent also no sugar or no antigen. So this is the only possible genotype in the case of blood group O. They don't have any antigen, no antigen or antigen B. So these are the different blood groups or the blood types of the progeny. So there are many questions they may ask based on the blood group typing, right? So if the parents are having the blood group that is A or B, then what will be the possible blood type of the offspring? So these type of questions we must be able to answer. So you're clear with this, right? About the blood type or the phenotype. So any doubts on this? Are you clear with this? So the next is, so maybe in the end, we'll see a few problems related to this blood grouping. Okay. And the next non-Mendelian inheritance is the multiple allelism. So this blood group, ABO blood grouping, is also an example for multiple allelism. So what do you mean by multiple allelism? It is the presence of more than 2 alleles of a gene to govern some character. So till now, we saw that only 2 alleles of a gene, they govern a particular character. But here, if you take the ABO blood grouping, 3 alleles are present, that is IA, IB, and I. But since humans are diploid, only 2 alleles are present in an individual. So they are diploid. So in an individual, only 2 alleles are present. So any 2 alleles may be present in a particular individual, and multiple alleles can be found in a population. So if you take a population or the many people, right, we can find the multiple alleles. For example, one person may have IA, so the blood group will be A. So this person blood group will be A. So the another person will have IA and I. So here also the blood group will be A. And some other person will have IB and IB. So the blood group will be B. So here B and A, B. So the blood group is AB. So if I is present, there's no antigen. So the blood group will be O, right? So each individual will have only 2 alleles, but multiple alleles are found in a population, right? So these alleles are present in different individuals. So, but 3 alleles, they control the ABO blood grouping. Then next is called the polygenic inheritance. So what do you mean by polygenic inheritance? It is the inheritance in which some traits are controlled by several genes. That is, multiple genes control one trait. For example, if you take the human skin color or the human height, we can see that the parent will be of some color or height, and the offspring will not be exactly equal to that of the parents. And one thing about the Mendel's law of inheritance is he didn't say anything about the blending of characters. So what do you mean by blending of characters? He crossed the pure tall and the pure short, and he found that in the heterozygous form only the offspring resemble the parent, right? And he didn't observe any of the intermediary characters between the tall or the short. So that is one of the disadvantages in the Mendel's law of inheritance. So he didn't observe any of the intermediate characters. So he didn't express anything about the blending of characters, right? So here in the case of polygenic inheritance, if you take the human skin color or the human height, so there occurs a variation. Not all the individuals are of same height, or not all the individuals are of the same color. So here there occurs a variation. Right? So it also considers the influence of the environment. Environment also, it influences the color of the person and also the height. So in the case of polygenic trait, the phenotype, it reflects the contribution of each allele. So there are many alleles which is responsible for the skin color or the human height. And the effect of each allele is additive. So they together contribute to the skin color or the height. For example, if you take the color of the skin, if 3 genes, for example, A, B, C, they control the skin color. So here the dominant forms are the responsible for dark skin color. So if the person is having all the dominant alleles, then the color will be dark. And the recessive forms, small a, small b, and small c, therefore light skin color. So genotype with all the dominant alleles will give the darkest skin color, and genotype with all the recessive alleles will give the lightest skin color. So the genotype with 3 dominant, so among these 6, if 3 dominant is present and 3 recessive alleles, it gives intermediate skin colors. So all these are additive, that is by the addition of the individual effect of these alleles based on how many dominant and how many recessive alleles are present. In this, the number of each type of alleles in the genotype, it determines the darkness of the lightness of the skin. So this is called the polygenic inheritance. So what do you mean by polygenic inheritance? It is the inheritance in which the trait are controlled by several genes or multiple genes are responsible for the inheritance. Then that is called the polygenic inheritance. Okay. Then the next is called the pleiotropy. So what do you mean by pleiotropy? That is a single gene. The single gene itself, it exhibits the multiple phenotypic expression. So that shows that is responsible for many phenotypic effects. That is for example, if you take some color and also the size of the seed, everything is controlled by a single gene. Because a single gene, it exhibits the multiple phenotypic expression, then the gene is called a pleiotrophic gene. Okay, so there are many examples. That is the mechanism of pleiotropy is the effect of a gene on a metabolic pathways which contributes towards different phenotype. For example, if you take the sickle cell anemia or phenylketonuria, so what happens in that is it will, because of the effect of the gene that is defect in this, because it affects the metabolic pathway. As a result of it, for example, if you take the phenylketonuria, it causes mental retardation and reduction in hair and skin pigmentation. Because of the defect in the metabolic pathway, it contributes to many phenotypes. So all these are due to the defect in the single gene. Okay, the phenylketonuria. So similarly, in the case of sickle cell anemia also. So the mutant gene has many effects. It shows many prominent effects, phenotypic effects, that is the physical appearance. Okay. So examples for the pleiotrophic genes are starch synthesis in P, phenylketonuria, and sickle cell anemia. In the case of phenylketonuria and sickle cell anemia, the mutant gene has many phenotypic effects. Example, the phenylketonuria, it causes the mental retardation and also reduction in the hair and skin pigmentation. But if you take the starch synthesis in P, that is this starch synthesis of starch is effectively, that is starch is synthesized effectively by homozygous dominant BG. So this BB is responsible for the synthesis of starch. So if the plant has homozygous dominant B gene, then large starch grains are produced. So it is responsible for the production of the large starch grain. That is the size of the starch grain. It influences the size of the starch grain. Okay. And if you take the homozygous recessive, they have the lesser efficiency in starch synthesis. So they produce smaller starch grains. So whether the starch grain is large or small is determined by the dominant or the recessive nature of this allele. Okay, but the starch grain size also shows incomplete dominance. So what do you mean by this? That is, if they are crossed together, that is BB is crossed with the homozygous recessive, that is small size. The F1 generation, the heterozygous form, it shows the intermediate size. So this shows the presence of incomplete dominant. So if it is completely dominant, so what will be the heterozygous here? The BB will be, so the B will be dominant, and here in the F1 generation, it will be large size, correct? So that is as per the Mendel's law of dominance. Here, we won't get the intermediate size. So the presence of intermediate size in the F1 generation, it shows the presence of incomplete dominance. But the same B gene is also responsible for the shape of the grain. For example, if it is present in the form of BB, that is homozygous dominant, it shows the round shape of the, after maturation, it shows the round seeds. And if it is present in the form of a heterozygous form, they also shows the round. And in the case of homozygous recessive, it is wrinkled. So here B is the dominant allele here. So as B is the dominant allele, here what happens? It shows the same dominant character in the F1 generation. So here the same B is a dominant allele. That is, it shows the law of dominance, dominant allele. But here, when we compare it to the size of the starch grain, here it is incomplete dominance. Okay, so the same B gene is dominant for a particular character and it shows incomplete dominance for a particular character. So dominance is not an autonomous feature. So if B, that is homozygous dominant B is present, it is not that it is dominant always. So it differs for the particular phenotype. So as far as the shape of the seed is concerned, it is dominant. And as far as the size of the that is seeds that are produced, then it shows incomplete dominance. Okay. So what do you mean by pleiotrophy? So pleiotrophy is the single gene. It exhibits the multiple phenotypic expressions. So that is called the pleiotrophic gene. Okay. So you're clear with this, right, till now? So all these will come under non-Mendelian inheritance. That is, we saw about incomplete dominance and then codominance, what are multiple alleles, polygenic inheritance, examples of the skin color and the height, and then about the pleiotrophy, that is the single gene which it exhibits the multiple phenotypic expression. It is called a pleiotrophic gene. Okay. So next, we'll see about the chromosomal theory of inheritance. So what do you mean by chromosomal theory of inheritance? So here, when Mendel did a lot of work on the pea plants and his work remained unrecognized. Why it was? Because he published his paper, his work in the journal, right? But the communication was not easy in those days, so it didn't reach many people. And also he used the mathematical approach, which was new and it was unacceptable in those days. That is, the mathematical approach in biology was not acceptable. And also the concept of genes, and he referred it as factors actually, as stable and discrete units, could not explain the continuous variation. So what does this mean? There occurs and many variation in an environment. You can see the N variety of plants, right? So many varieties, but he couldn't account for the variation that exists in the organisms, because he said that only it will look like a parent and there is no blending or the mixing of the characters, right? So he didn't explain anything about the variation. And also he could not give the physical proof for the existence of the factors or genes. So in 1900, that is after when he was alive, there was no recognition. Only later in 1900, that is Debris, Correns and von Schermack. They are from different places. That is Correns from Germany and he's from Holland, and von Schermack from Austria. They worked independently and they rediscovered the Mendel's results. So they did more work on the Mendel's law of inheritance. So next, the chromosomal theory of inheritance was actually proposed by Walter Sutton and Theodore Boveri. It was proposed in 1902. So what they proposed actually during this time, due to the advancement in the discovery of the microscope, and they were also able to observe the features that are present in the nucleus and about the cell division. So they were able to say that they were able to observe the nuclear division and the meiosis, right? So they said that the pairing and the separation of a pair of chromosomes, it lead to segregation of a pair of factors they carried. So here what they said, pairing and separation of a pair of chromosomes occurs. So in the chromosomes, so they discovered about the chromosomes, right? And also they studied about the meiotic division. So they said that separation of a pair of chromosomes, it leads to the segregation of the pair of factors, that is the genes, right? So he united the chromosomal segregation with Mendelian principles. So both chromosomes and genes, they occur in pair, and he linked the chromosomal segregation with that of what Mendel said as factors or the genes. So, and he called it as the chromosomal theory of inheritance. So he linked it with the chromosomal segregation. Okay. So what he said, that is the chromosomes are the vehicles of hereditary. So they are responsible for the hereditary or the transfer of information from parent to progeny. And two identical chromosomes, they form a homologous pair. So because they form a homologous pair, and this homologous pair, it segregates during gamete formation. So these are the main steps. Actually, we saw everything under cell division, right? So here during the anaphase, particularly during the anaphase, there occurs a segregation or the separation, right? So the homologous pair, it segregates during gamete formation. So these are the gametes that are formed, and independent pairs, they segregate independently. So this is what that is under the chromosomal theory of inheritance. That is, they linked the chromosomal inheritance. That is, they united the chromosomal segregation or the separation in the formation of gametes with Mendelian principle. Okay, so this shows the formation of the germ cells. That is how they separate, or how the independent pairs they separate and they form the gametes. So then Thomas Hunt Morgan, he worked with the fruit flies to prove this chromosomal theory of inheritance. So with the fruit flies, that is the Drosophila melanogaster. So why he chose the Drosophila melanogaster is they can grow on a simple synthetic medium, and they have the short generation time, that is life cycle is only about 12 to 14 days, so it can be easily studied, and breeding can be done throughout the year. So you can easily use for experimental studies, and it produces hundreds of progeny per mating. So there will be more availability, right? And even male and female flights are easily distinguishable. That is, male is shorter. This is a male is shorter than the female. So they are easily distinguishable, and there's many types of hereditary variations that can be seen with low power microscope. So it is easy to study all the factors. So Morgan chose the Drosophila melanogaster to prove the chromosomal theory of inheritance. So what he did is he carried out several dihybrid processes. That is, he chose what is a dihybrid process? He chose the two different characters, right? He's carried out the several dihybrid processes in Drosophila to study the sex-linked genes. So what do you mean by sex gene? That is, if they are associated with the X and Y chromosome. Okay. So in cross 1, what he chooses, yellow-bodied, white-eyed females are crossed with brown-bodied and red-eyed males. So this is the wild type gene, and he crossed both of these. And in cross 2, so he took the two different varieties, and here he took the white-eyed miniature winged, which is crossed with red-eyed and large winged. So these are crossed together. So when he observed it, what did he observe? So this is the cross actually, right? So in the cross A, that is here. This is the cross A parental generation and this is the wild type. And in the cross B, he chose the white and the miniature, and here it is yellow and white. So when he crossed, in the case of cross A, the recombination is only 1.3%. But in the case of cross B, he found the recombination as 37.2%. So how that happens? That is, the two genes did not segregate independently, and the F2 ratio actually deviated from the normal ratio of 9:3:3:1. This is the dihybrid ratio, that is as per the Mendel's law, right? Dihybrid ratio is 9:3:3:1. But these two genes, they did not segregate independently. As per the Mendel's law, they know that they segregate independently, but here it didn't happen like that. Okay, and the F2 ratio deviates from the ratio. The genes were located on the X chromosome, and when two genes were situated on the same chromosome, proportion of the parental gene combinations was much higher than the non-parental type, and this is due to linkage. So what do you mean? If you see the cross A, so here they are found closer together, but in the case of cross B, they are found somewhat separated, right? The distance is higher in compared to that of the cross A. So here, when the genes are situated on the same chromosome and when they are so close together, there will be linkage, right? So there will be a linkage, and the proportion of the parental gene combination was, that is, they are linked together, so they exist in the form of parental gene combination, and the recombination is less in the case of linked genes. So the linked genes are actually located on the same chromosome, right? So there are two types of linkage. One is called the complete linkage and another one is called the incomplete linkage. So it's the case of complete linkage. So what is complete linkage? Here the grouping of genes or the linking of genes on a chromosome which is not altered. Grouping of genes on a chromosome is not altered. And is inherited as such. And is inherited as such. So there is no change as such. Okay. So from generation to generation, they will be inherited as together. So there will be no change. But if you take the incomplete linkage, so here there occurs a crossing over between two homologous chromosomes. Between two homologous chromosomes. Or one or more alleles present in the linkage group, they are replaced by the another allele. So if you see here the recombination. So this is the recombinant thing. We can see the blue and yellow together, right? So here there occurs a recombination, but the recombinant type is very less. But here if you observe, the recombination type is very less as the genes are located far together. Okay, so when they are close together, they will be inherited together. So they are called linked genes. So grouping of genes on the chromosome is not altered and they are inherited as it is from generation to generation. But in the case of incomplete linkage, the crossover occurs between the two homologous chromosomes, so that one or between the two homologous chromosomes, so that one or more alleles present in a linkage group or replaced by another allele. replaced by other alleles. Okay, so is it clear? So this is called incomplete linkage and this is a complete linkage. So here if you observe, when two genes were situated on the same chromosome, proportion of parental gene combinations was much higher than the non-parental type, and this is due to linkage because they are linked together. So if you see the genes of white eye, yellow body, they were tightly linked because they are close, right? So they are, this is the case of yellow and white. So they are closer and they are tightly linked. So they are called the linked genes. So they are tightly linked and showed only 1.3% of recombination. But if you take the genes of white eye and miniature wing, they are loosely linked. There is not much linkage and they show 37.2% recombination. Okay. So this is the conclusion, that is tightly linked genes show low recombination and loosely linked genes, they show high recombination. Okay. So this was explained by the Morgan's experiment. So tightly linked genes, they show low recombination and loosely linked genes, they show high recombination. So I hope you are clear with this, right? So this is the complete and incomplete linkage. So then Alfred Sturtevant, he used the recombination frequency between the gene pairs for measuring the distance between genes and mapped their position on the chromosome. So based on the recombinants that are formed, so by means of the crossing, based on the recombinants that are formed, he found out the distance between the genes. So what is the, for example, if more number of recombinants are formed, then what does this indicate? In the previous case we saw, right? So here it shows the formation of more recombinants. So the distance is greater. So here the less recombinants are formed. So the genes are closely, they are closer, right? So based on the recombinants that are formed, the distance between the genes can be measured and can map their position on the chromosome. So where the genes are located on the chromosomes can be found out. So it was done by Alfred Sturtevant. So these genetic maps, they are used as a starting point in the sequencing of genomes. Example, in the case of Human Genome Project. Okay. So any doubts in this regarding the chromosomal theory of inheritance and linkage and variation? So any doubts? So we are clear with this. Okay, so the strength of the linkages. So the strength of linkage is actually it is proportional or inversely related to the distance between linked genes. Distance between linked genes. Okay. And also it is proportional to 1 by crossing over. So what is genetic map? Actually, it tells us the position of a particular gene. Position of a particular gene on a chromosome. Particular gene on the chromosome can be found out using a genetic map. That is, the position of a particular gene on a chromosome can be found out by using a genetic map. So, for example, we'll discuss one problem related to the genetic map, that is how to find the distance. Okay. So, for example, in the case of maize, colored endosperm. So, colored endosperm is a dominant, that is C, right? It's dominant over colorless. That is C. C, it represents the colored endosperm. Okay. And C is dominant over C, which is colorless. Then R, that is full endosperm, which is dominant over shrunken. This represents the shrunken. Okay, so when a dihybrid of F1 generation was test crossed, it produced 4 phenotypes in the following percentage. That is, colored full is 48% and colored shrunken is 5% and colorless full, it shows 7% and colorless shrunken, it shows 40%. So from this data, what will be the distance between the two non-allelic genes? Distance between the two non-allelic genes. So the options are 48 units and 5 units, 7 units and 12 units. So any idea of how you will find out the answer for this? That is, in maize, colored endosperm is dominant over the colorless. So what is the correct answer for this? So what is the answer? So colored endosperm is dominant over colorless and the full endosperm is dominant over the shrunken. And when a dihybrid F1 generation, so F1 generation will be heterozygous. When it is test crossed, it produces the 4 phenotypes in the following percentage. So distance between the two non-allelic genes, it is asked. So what will be the answer? Nandini, you are able to follow?
Yes, ma'am. Yes, ma'am.
Yeah. So what will be the answer for this? So here one map unit is, so what is one map unit? Is
1
that,
Morgan.
yeah, one, what is 1 Morgan or 1 centimorgan? Is the distance that yields 1% of recombinants of recombinant chromosomes. So what is the answer for this? So here, what is the percentage of the recombinants that is formed in this question? So what is the percentage of recombinants that is formed? For example, the colored full is the parental. So we are crossing the colored full with the colorless shrunken. So this is a parental generation and also colorless shrunken is the parental generation. So here the recombinants are colorless full, that is 7% are formed, and colored shrunken 5%. So these two are the recombinants. So 12% on the, so it is it produces about 12% recombinants, right? So here one map unit or the 1 centimorgan is equal to the distance that yields 1% of the recombinant chromosomes. So here it is 12%. So the answer is 12 units. So, right, are you able to follow this? So the answer is 12 units because the distance that yields 1% recombinant chromosome is the 1 centimorgan, and 100%, it is 100% crossover, it is equal to morgan. Okay, so the answer is 12 units.
Ma'am, explain again, ma'am.
What you want me to explain?
Yes,
That is, if you, the question is, we are crossing the colored endosperm with colorless shrunken. So here colored endosperm, colored and the full endosperm is a dominant character, and colorless and shrunken are the recessive characters. So when a dihybrid of an F1 generation, so dihybrid is both of them will be a heterozygous form, right? RR. So when it is crossed with the recessive, it was test crossed. That is when it is crossed with the recessive parent, we are getting in this percentage. That is colored full, we are getting 48%, and colored shrunken 5%, colorless full is 7%, and colorless shrunken is 40%. So among these, if you observe it, here the colored full, this is of the parental type. It is colored full, and colorless shrunken is also of parental type, right? Because we are crossing these two. But these two are the recombinants that are formed. That is colorless full and colored shrunken are the recombinants that are formed. So the question is, what will be the distance between the two non-allelic genes? So here we have to calculate the total percentage of recombinants that are formed. That is 7 and the 5%. So total recombinants is 12%. The recombinants that are formed is 12%, right? So here the one map unit is equivalent to the distance that yields 1% of the recombinant chromosome. So here the 12% of recombinants are formed. So here the answer is 12 units. So based on the percentage of the recombinants that are produced, we can find out the distance between the non-allelic genes. Okay, are you clear with this now? Is it clear? So shall we move on to the next question? So any doubts? Yes. So we'll discuss few questions. So among the 7 pair of contrasting characters or the contrasting trait in pea plant studied by Mendel, the number of traits related to flower, pod, and seed respectively was option 1, 2, 2, 2, and option B 2, 2, 1, and option C 1, 2, 2, and option 4 1, 1, 2. So what is the right option? Yes, A is the right answer, because the flower, that is which is related to the flower, is 2, that is color and the position of the flower, and which is related to the pod is also color and shape, and related to the seed is color and shape. So 2, 2, 2 is the right answer. The color-based contrasting traits in 7 contrasting pairs studied by Mendel in P were 1, 2, 3, and 4. So what is the right option? The color-based contrasting traits in 7 contrasting pairs studied by Mendel in pea plants were 1, 2, 3, and 4. 4. That is color-based. So what are the characters he took for color? The color-based contrasting traits in 7 contrasting pairs. So he took 7 contrasting pairs, among that color-based pairs. So how many are color-based? The options are 1, 2, 3, and 4. So it's not 4, actually it is 3, because he took into account of the color of the flower, pod, and the seed. Only 3, right? Flower, pod, and seed. So only these 3 are the color-based. The others are position of the flower, and then shape of the pod, and then shape of the seed, and then stem height, tall, and the dwarf. So only 3 are the color-based traits, right? So 3 is the right answer. So clear? So Mendel observed that all the F1 progeny plants A resembled either one of the parents, resemble neither of the parents, resemble both the parents, and shows 3:1 ratio. Mendel observed that all the F1 progeny plants resemble either one of the parents, resemble neither of the parents, resemble both of the parents, and it shows 3:1 ratio. In the F1 progeny, he asked, yeah, A is the right answer because he asked in the F1 progeny. So F1 progeny, it resembled either one of the parents, right? So according to Mendel, factors or genes or the units of inheritance, they contain information that is required to express a particular trait, both 1 and 2 and none of the above. Yeah, C is the right answer. Then the phenotype of any character will not be affected if the modified allele produces normal enzyme, non-functional enzyme, no enzyme at all, 2 and 3 both. The phenotype of any character will not be affected if the modified allele produces a normal enzyme, non-functional enzyme, no enzyme at all, and then 2 and 3 both. Yeah, A. So if it produces a normal enzyme, the phenotype of any character will not be affected, right? So A is the right answer. And which cross yields red, white, and pink flowers variety of dog flower? So the combinations they have given is, the first is homozygous dominant, this is heterozygous, and option B, it is heterozygous with homozygous dominant, and option C is both heterozygous. Then this is heterozygous with homozygous recessive. So which cross yield red, white, and pink flowers? So all the three should be there. So you check by putting the Punnett square so that we can find out, right? So which cross yields red, white, and pink flowers? So what will be the option? Yes, C is the right answer. That is RR. Only here. So if you see the A, what will happen? Only possibility is RR and heterozygous, right? So here also RR and heterozygous, there will be no formation of the homozygous recessive. So only in this combination here you will get the RR and also heterozygous and also recessive. So if you observe this cross, it will be either heterozygous or homozygous recessive. So only in option C you will get all three type of flowers. This will be of red, this is white, and this is pink, right? So C is the right answer. Then which of the following is exception to Mendel's laws? Linkage, incomplete dominance, codominance, all of the above. Which of the following is exception to Mendel's law? Is D, all of the above. All are an exception to Mendel's law. Then in Mirabilis and Antirrhinum plant, the appearance of pink hybrid between cross of a red and white flower parent indicates incomplete dominance, segregation, dominance, and heterosis. Yes, incomplete dominance. The recessive characters are option A, only expressed in heterozygous condition, only expressed in homozygous condition, blend in heterozygous condition, always impure. The recessive characters are only expressed in heterozygous condition, only expressed in homozygous condition, blend in heterozygous condition, always impure. No, it is not option. Yes, it is option B. Okay, the recessive characters are only expressed in the homozygous condition, right? It is not option C. In option C is blending, but blending will not happen in the case of recessive characters, right? So that is not the correct answer. In case of incomplete dominance, the monohybrid ratio of phenotypes in F2 generation is 1:2:1, 3:1:1, 9:3:3:1, 2:3:1. Yes, it is option A, that is 1:2:1. Then, when the phenotypic and genotypic traits resemble in the F2 generation, it is an example of independent assortment, qualitative inheritance, segregation of factors, incomplete dominance. Yes, it is an example of incomplete dominance, because in the case of incomplete dominance, phenotype and genotypic ratios, they resemble, both will be the same. Then, which of the following conditions represents a case of codominant genes? Option A, a gene expresses itself suppressing the phenotypic effect of the alleles. And option B, genes that are similar in phenotypic effect when present separately but when together interact to produce a different trait. And option C, allele both of which interact to produce a trait which may resemble either of the parental type. Option D, alleles each of which produces independent effect in heterozygous condition. So which is the right option? In the case of codominant genes, so what will happen in codominant? Option C means it is given as that is allele both of which interact to produce a trait which may resemble either of the parental type. So that is not happening in the case of codominant. Codominant, actually both the genes, they will express together, right? Here they may resemble either of the parental type it is given. So that is not the right answer. That is option D is the right answer. That is alleles, each of which produces an independent effect in heterozygous condition. That is, both will have an independent effect. For example, if you take the blood group AB, it will produce A antigen also and B antigen also. That is, it produces an independent effect. Both the effects will be there and both of them will be expressed. Okay, so option D is the right answer here. Yes, the next question. So Mendel did not propose dominance, incomplete dominance, segregation, independent assortment. So just the, yeah, B is the right answer. Then ABO blood group is an example of epistasis, multiple allelism, pleiotropism, complementary genes. Yes, it is an example of multiple allelism. So if child is blood group, the child's blood group is O. The blood group of the child is O. His parents' blood group cannot be. Option A, B and O. Option B, it is A and O. And option C, it is AB. And option D, it is A and B. So if the child is having an blood group of O, his parents' blood group cannot be. So which is the right answer for this? C is the right answer. It cannot be AB. So it can be B. That is B. It may be of IB and so it can be of either IB or I. Right? So this I and here it will be II. So it can be either A and O also, but AB cannot be, because if the parents are having the AB, the child should have either A or B, right? So C is the right answer. And if one parent has blood group A, the other parent has blood group B, the offsprings have which blood group? The options are AB only, then option B is O only, B only, and here it is A, B, and O. So the parent blood group of A and B. So which is the right? Yes, D. So if they have the A and B, the child have either A or B, or the child can have A, B or O. So D is the. So I think you are clear with this, right? About the blood grouping. So if the parental blood group is given and if they ask you to find out the child's blood group, you should be able to answer. Okay, you're clear with it, right? So, and also if the child blood group is given, and you must be able to find out the parental blood group. Okay. Then the ratio 9:7 is due to supplementary genes, lethal genes, complementary genes, and epistatic genes. The ratio 9:7 is due to supplementary genes, lethal genes, complementary genes, epistatic genes. Yes, it is C. That is complementary genes. We saw yesterday, right? 9:7 is complementary genes, and for supplementary genes is called 9:3:4. And in the case of epistasis, that is dominant epistasis, it is 12:3:1. And supplementary genes is also called the recessive epistasis, which is also called recessive epistasis, and the ratio is 9:3:4. Okay. And when two independently assorting dominant genes interact with each other to produce particular phenotype, but when they present alone, they did not produce phenotype, they are called complementary genes, supplementary genes, duplicate gene, and inhibitory gene. So when two independently assorting dominant genes, they interact with each other to produce a particular phenotype. But when they are present alone, they did not produce any phenotype. When they are together, they will produce an effect. But when they are alone, when those alleles are alone, they did not produce any phenotype. And they are called complementary gene, supplementary gene, duplicate gene, and inhibitory gene. So the correct answer is option A, complementary gene. That's why we got the ratio of 9:7, right? So when they are together alone, they'll produce the effect. But when they are present alone, they did not produce any phenotype. So complementary gene is the answer. You go through those definitions. Yesterday we saw, right? Under complementary genes, supplementary genes, and epistasis. So definitions and ratio, you be clear with that. So if any questions may come on that, okay, you can answer. So any doubts in this till now? So we have completed the monohybrid cross, then dihybrid cross, and about the non-Mendelian inheritance. So under that, intragenic interaction, intergenic interaction, and chromosomal theory of inheritance, and how linkage and recombination. So if they are present in the linked form or the linked genes, then the possibility of recombination is less. So based on the number of recombinants that are formed, we can find out the distance between the genes, right? So that is expressed as the centimorgan. So you go through that also. So based on the number of or the percentage of recombinants that are formed, we can find out the distance between the genes, right? So based on the distance between the genes, and we can find out the position of a gene in the chromosome. That is the genetic map. Okay. So, and then tomorrow we'll continue the next topic. Okay, sex determination, and then about the mutation. So clear, right? Any doubts in this? And you go through some of the problems, because as far as the principles of inheritance and variation is concerned, you have to be clear with the most of these problems. And it is by means of crossing these two characters. So what will be the character in the offspring or the progeny, and also the percentage of the offspring that is produced. So practice these type of problems in this chapter. Okay, and also under blood group, what are the possible blood types that can be found in an offspring if the parents are of these two blood group. Okay, so those type of questions also you practice in this chapter. Clear? Okay then. So we'll continue tomorrow. Okay, thank you.